Hi, welcome to my poster. My name is Jiahui. I am a postdoc in the Department of Biomedical Informatics, University of Pittsburgh. Here I am presenting our work on aberrant alternative polyadenylation landscapes in end-stage liver disease. Alternative polyadenylation, or APA, is a phenomenon where the different usage of poly-A sites can produce different mRNA isoforms. In a simplified case, using a proximal PAS can lead to mRNA with shortened 3' untranslated region. It has been found that a longer 3' UTR is associated with reduced mRNA stability and gene expression. Although most polyadenylation happens in the 3' UTR, you can also use PAS as the in-chain region. Intronic polyadenylation has multiple subtypes, including the composite IPA, where the terminal axon is formed by an internal axon and an extension part from its adjacent in-chain. The other type of IPA is called skips IPA, where a hidden alternative axon is used as the terminal axon. As we can see, IPA could result in changes in the coding sequence and produce truncated mRNA, which can further lead to dysfunctional proteins or reduced gene expression. Changed polyadenylation patterns have been implicated in various diseases. It has been suggested that dysregulated APA contributes to dysregulated metabolism in fatty liver disease. End-stage liver disease involves extensive fibrosis and the loss of hepatic function, which can further lead to liver failure. While most studies focus on coding mutations or gene expression levels, we wanted to see how RNA processing, especially the APA, might be disrupted in ESLD. To systematically characterize APA and IPA alterations in hepatocytes from end-stage liver disease patients, we used bulk RNA-seq data from human hepatocyte samples from individuals with and without ESLD. Here we used a method called DPAS to examine the dynamic APA events and used the IPA finder method for detecting the IPA events. The advantage of these two methods are that they can use standard bulk RNA-seq data to identify poly-A sites without annotation reference. These two methods model the RNA-seq coverage and infer poly-A sites by identifying where the read coverage suddenly drops. DPAS first predicts the location of the proximal poly-A sites, then calculates the percentage of distal poly-A site usage index, also called PDUI, to estimate the proportions of transcripts using the distal versus proximal poly-A sites. When comparing two conditions, samples with lower PDUI values have increased usage of proximal poly-A sites, producing transcripts with shortened 3' UTR. Similarly, IPA finder quantifies the relative usage of poly-A sites for a gene between conditions. Samples with higher IPUI values have increased IPA events, indicating increased production of truncated mRNA. In our analysis, we identified 261 significantly altered 3' UTR APA events across 163 genes, with a strong bias towards shortening. Among these, 22 genes, including several liver disease-related genes, have shortened 3' UTR, and they also have increased expression in ESLD. IPA finder identified several metabolism-related genes with increased IPA events and also accordingly reduced expression in ESLD. For example, from the coverage plot here, we can see the 3' UTR shortening for the gene VEGFA. This is the gene that has been associated with fibrosis progression. Another example for IPA event changes in ESLD. Here we can see the increased composite IPA events in ESLD for gene LRP1. This is a metabolism-related gene that could contribute to ESLD progression. To further explore the potential mechanisms underlying these polyadenylation changes, we evaluated the expression of APA factor genes. We observed altered expression of key APA regulatory RNA binding proteins and splicing factors in ESLD, including the downregulation of CPSF3 and SRSF1, which may contribute to these observed APA defects in ESLD. In conclusion, ESLD shows dysregulations in APA and IPA in several genes that are very important in liver function and metabolism, revealing new opportunities for mechanistic insights and therapeutic developments. These results could potentially be improved with larger sample size in the future. So that's all for my poster. Thank you for your attention. Please let me know if you have any questions.